wait 5 minutes and start in 3 and 5 minutes, so everyone has the chance to join. And really looking forward to the webinar. So 5 more minutes to wait. Just a few seconds left, and then we can start, I guess. A lot of people already joined. So I think we can start. Hi, Max, first of all.
Hello.
And everyone else. Thank you for joining the Permeability series, the first one of the year. But yeah, as you might know, Fabio just was founded this year, so we had to start the series again. And I'm very happy that we continue with this very educational series and with Max as our presenter. And Max will present the topic commercially available cell-free permeability tests for industrial drug development, increased sustainability through reduction of in vivo studies. And as you don't know, my name is Yannick, and I will be your host for this webinar today. And on today's agenda, we will take a closer look on tools and barriers that help you to investigate the permeability of drugs. From the basic questions, why to use in vitro tools to predict in vivo performances, to the explanation of an important indicator, the apparent permeability, and concluding with the practical examples of experiment setups. We are happy to again have Max as our presenter. He has a very impressive academic vita, total expert in pharmaceutics, and especially in drug formulation and permeability. Next to that, Max is also a freelancing scientific consultant for Fabiok in Germany, the company which commercializes PermaPet. Max is the co-inventor of PermaPet at the University of Southern Denmark. Before we start the webinar, a few things regarding the organization. We have opened a Q&A window on the right-hand side where you can type in your questions every time, and we will answer them after the presentation. And we also will provide the slides and the link to the recorded webinar at the end of the presentation or in the next few days. Without further ado, I give the word to Max. Enjoy the presentation and see you at the Q&A session again. Thank you.
Thank you very much, Yannick. I hope you can all hear me well.
Yes.
Okay, then I'm sharing my screen, and I just would like a feedback. Is the presentation visible?
Yes.
Okay. Okay, thank you. Thanks, Yannick, for the nice introduction. And thanks also for the audience of this webinar. Today, I will present you. I will make a presentation on commercially available cell-free permeability test to increase the sustainability through a reduction of in vitro studies. And as you can see, the title is basically the same of a recent review paper. We have I wrote with several other colleagues from other university in Europe, describing these types of tests and why they are useful in early stage drug development. So the outline of the presentation, I will start presenting the sustainability issue of early stage drug development. I will present the commercially available biometric barrier and permeation setups. And I will conclude presenting the more advanced dissolution permeation setup. So instrument and apparatus that can both give you an indication on the dissolution of a solid dosage form and the permeation of the API after dissolution. So, as a very general, to begin the presentation in a very general, with a very general perspective, let's have a look at the drug development journey. So, this is the drug development from discovery up to commercialization can take up to 10 to 12 years in general, and is generally divided in 4 different steps. The first steps is the so-called drug discovery, where actually the chemical entities are identified and the potency on the receptor on the pharmaceutical and therefore the pharmacological effect are measured. And this is the part also where the so-called drugability properties are measured. So mostly the solubility of the compounds. In former times, it was very useful, the partition coefficient, and nowadays, the permeability. After this stage that can last from 3 to 5 years, we have the preclinical stage where actually they identify the compounds, the promising compound, let's say a panel of 250 to 500 compounds, are formulated into prototype of formulation and also tested in vitro testing, in vivo testing, ADME, PK, PD studies. Of course, these are the parts where animal testing is still heavily used, especially at industrial level. After this, we have the clinical trial divided in 3 phases. Also, this is also kind of time consuming and cost consuming. And in let's say that from as a rule of thumb, from 5,000 to 10,000 compounds, the pharma industries can generate one new drug with a total expensive of, yeah, this is vary a lot, but goes from 1 to 1.3 billion euro. So as said, the preclinical part, including also drug discovery, is the one that is definitely utilized in the most animal, non-human animal testing. And so, which is the impact of this type of studies? Well, let's say that let's say that in Europe, at least, the use of animal studying is definitely not declining. This is data from the European Commission that stops in 2018, and so what we can see is that number of animal testing is definitely not declining. It in Europe is around 10 to 12 million animal testing per year, and of course worldwide we should multiply this by a factor of minimum 10. Uh, and how sustainable are these animal testing? Well, animal testing indeed are much less sustainable than standard lab. If we look, for example, of average water consumption, animal lab facilities consume circa 4 times more water than a standard lab. And when it comes to electrical consumption, this value goes 20 times higher. And why that? Because animal facilities require a lot of energy because animals need to be kept at a constant room, at a constant temperature, and at a constant humidity level. And these, of course, require a lot of energy. So it would be much better also from a sustainable point of view to convey into new in vitro tools to substitute this kind of studies. And one of the parameters that has been around for the last 20 years that could actually be used to substitute the in vivo bioavailability measured in animal after administration is the permeability or the upper permeability coefficient. The permeability was actually permeability was not used as a word in pharmaceutics before 95, when actually Amidon introduced the BCS classification system, disclosing the so-called human permeability. After this, some just 3 hours later, 3 years later, Kenzie et al. Introduced the first in vitro permeability tools, the PAMPA, a parallel artificial membrane permeability assay. And just 3 years later, Lipinski introduced the so-called rule of five that we all know. The combination of these three effects has definitely boosted permeability as a concept because the number of publication and citation has exponentially grown after these times. So when actually was this concept introduced, we can say that the first description of the permeability was done by Amidon, which introduced actually the so-called human permeability. So the permeability of a compound through the gastrointestinal mucosa, and but that parameter was not an in vitro parameter, something that could be measured with in vitro tools, empirical tools, but it was a parameter that was derived by in vivo data. Basically, the fraction of dose absorbed normalized over the surface area of the GI tract that can, from which they calculate the flux. And the concentration was defined as the concentration of the drug in the GI tract, or to be more specific, at the membrane intestinal wall surface. Uh, Making this calculation, it was possible for them to establish a correlation between this so-called human permeability and the fraction absorbed. So what we try to do with the upper permeability is to substitute the human permeability that is, and I repeat, an parameter that is extrapolated from in vivo data to a more in vitro parameter that we can actually measure empirically. For doing that, we need a biomimetic barrier. The classical biomimetic barrier were based on biological materials. Uh, I think all of you are familiar with the CACO2 model developed by Artuson in 96, where actually a monolayer of cells are of intestinal adenocarcinoma cells grow on a filter support, and they are used to study permeability of drugs and active ingredients. The episkin is also a cellular model, but that reassembled skin. And then we have the classical ex vivo tissues. For example, here you can see the ear of a pig that have been used widely and still are used for skin studies. When we talk about cell-free biomimeric barriers, so barriers that are biomimeric, so reassemble the permeability of biological tissue when interacting with the compounds, we have the PAMPA, the first introduced, the parallel artificial membrane permeability assay. We have the PermePed, that is the product commercialized by Fabioc, and that I also contribute to invent. And then we have two two membrane that can be used, but they are not, especially this regenerative cellulose, they cannot be considered biomimetic, but more like a size exclusion barrier. Transem is also is a multi-layer of lipophilic membranes that should reassemble the skin. The physical model of which permeability is built is basically the one represented here. Every permeability experiment is based on a donor compartment where the drug is placed, a barrier, one, and you can choose from one of the previous mentioned barrier, and the receptor compartment. The receptor compartment is basically where the analysis of the accumulation of the compound is measured by different methods, UV, HPLC, mass chromatography. And what we measure actually in this kind of studies is the flux, as defined as the amount of material Q that cross the barrier over time over the surface area of permeation, in this case the surface area of the barrier. Assuming that this diffusion or this movement of drug happens spontaneously due to a gradient of concentration and that are not significant interface, and of course this is an approximation, we can actually calculate the permeability dividing the flux measured by the concentration of the drug in the donor. The permeability is often expressed in centimeter per second, and in the case of Pampa, Permaped, and Cacochu is in the range 10 minus 5, 10 minus 6 in general. And there are generally two approaches for measuring. One is a very pragmatic one, the so-called endpoint. So incubating the barrier for like 4 to 5 hours and just measuring the permeability with one single point, or making a linear regression curve where we actually sample every, for example, half an hour, and then we replace the withdrawn volume with the fresh buffer to maintain the so-called sink condition. Of course, these two numbers will differ a little, but not significantly. One is, of course, much more rapid, and not more rapid, but it's much easier for the operator because you don't have to sample over time. And which kind of setups can we use? Well, we can use the classical vertical diffusion cells, for example, the front cell diffusion for Permegear or other providers, where actually we place the barrier within this small donor compartment and the acceptor compartment, where actually we sample from this neck. We also have horizontal diffusion cells, the side-by-side cells, Houssine chambers also can be provided by Permegear, but also other provider. But these, of course, are not very high throughput because we don't have the possibility to run much more than, for example, 8 to 10 parallels. That's why Permaped and also other barrier has been devised to be on a multi-well diffusion cell plate. In this case, this is the picture for the Permaped plates, where each of the well will represent a permeability experiment. So we can go up to 96 wells per plate. So this is definitely very much high throughput screening. And this is extremely good when we have to screen a lot of compounds. Focusing a little bit more about the composition and the structure of the barrier, the parallel artificial membrane permeability assay, the PAMPA, developed by Cancerol in 99, is basically a filter, inert filter, nitrocellulose or other sources, which is actually soaked in a solution of an organic solvent, octadiene in this case. This is the original recipe, and different type of phospholipids and cholesterol that and this combination should somehow reassemble the lipophilicity of the epithelium cells of the endothelium and the mucosal cells. So in this case, the drug molecules here represented by these blue dots permeate through the barrier mostly according to the partition or distribution properties. Um, on the other hand, the PermeaPad is a little bit I would say, an advancement in terms of biomimeric properties. This is how one pad looks like before it gets hydrated. The barrier is composed basically by phosphatidylcholine 100%, and upon hydration, in a matter of seconds, inside the barrier, a layer of phospholipid gels is spontaneously forming. So liposomes that press each other. And in this case, we have two mechanisms of permeation. One is based on partitioning, because of course of the phospholipid vesicles membrane, but also we have some sort of a percolation, so sort of a paraliposomal pathways that could be used that is used by small hydrophilic molecules, as well as would be the paracellular transport in a biological tissue. I named before that other type of membranes, like a regenerative cellulose, should not be really considered biomimeric. And here I reported an example of what I mean. So this is like an amorphous material, like fibers, that doesn't really have they have a cutoff, but they don't really have pores. And in this case, I have summarized the results of permeability measures utilizing this barrier in different works, some of my research or other published paper. And as you can see, the ranking of the permeability in this case goes much along with the molecular weight. So to say that with this barrier, we get more like a size exclusion of the barrier. So the permeability is linked mostly to the size of the molecule. Small molecule will permeate faster, large molecule will diffuse slower. But this is not really biomimeric, this is just diffusion based. That's why I believe, my personal recommendation is this barrier not biomimetic as such, but more classify according to the size. So just to have a small summary, but of course this is like a very simplistic conclusion, but the PermePad is extremely robust and has very nice biometric properties. Of course, all these barriers cannot account for active transport. As if it's in high throughput methods, and when we talk about also storageability, so storage, PermePad is really nice because it can be stored at room temperature without any issue, and its reproducibility is often very, very good. In general, superior than Pampa. The regenerated cellulose I also reported in this table, but as I said, this is not a biomeric barrier, and also the reproducibility is very low. The standard deviation is generally very high, and is not very suited for formulation testing. As also the pampa will be degraded most often by excipients, whereas the PermeaPad sustain testing of such enabling formulations. And I said before that the PermeaPad has this paraliposomal pathways, and this has been demonstrated in a publication, a very recent publication by Erickson and co-worker. They have made a very elegant study where they have modified the tonicity of the septored and Dorner solution in order to have the liposomal gel forming and with a different size of the liposome. So to allow the liposome to shrink or to swallow, and what they have been investigated using one of these compounds, caffeine, that the permeability of the compound was highly affected. So the permeability was much higher when the liposome shrinked, and it was much lower because of the swallowing of the liposome, proving that for some compounds, small hydrophilic compounds, as it is in real life, this PermePad can account also for these pathways of absorption and permeation. Especially with the old version of the pump, this was not very easy or very nicely detectable. For example, in an old version of Pampa, caffeine would have been detected as a medium-low permeable compounds, but this is not in agreement with what it in real life, because caffeine is one of the most bio available compound we have after oral administration, and that was and in PermePad this has always been read in a very correct way. And this is the latest overview of compounds. This is a set of 40 compounds that have been recently published, tested on PermePad, and this was done by Butnarhaus et al. At University of Turin. And as you can see, PermePad can discriminate nicely according a long range of permeation, and this analysis was done by mass spectroscopy. And in this work, they have selected a limit of 1.15 10^-6 to divide the drug from high permeable and low permeable. Of course, it has to be underlined that in this study, the concentration they used for this study was very low. Therefore, the absolute value of permeability can also vary according to that. And that was also done in an old version of the PermePad. Nowadays, the PermePad the permeability stretch up to 20 10^-6 centimeter per square. So this curve is much more distributed and heavenly distributed. And this is all the fitting of 16 compounds investigated by Jacobsen that actually were compared with the human fraction observed, and the permeability measure for PermePad was actually in good agreement with the human fraction absorbed reported in literature for the compounds tested. So what we have been discussing now is extremely useful for drug for when we test APIs or chemical entities or molecules, so to evaluate this so-called drugability. But of course, it is also nice to when we have a final formulation, especially a solid dosage formulation, to have an investigation of what is happening when we have also another steps, so the so-called dissolution. And for this reason, several dissolution permeation tests has been introduced. So tested, where actually some of them are based on the pharmacopoeia vessel, where you actually have dissolve your tablets, but you have also an insert where you actually that is actually equipped with a biomimetic barrier, and you can actually measure also instead not only the dissolution rate but also the amount permeate over time. And this should give a much better prediction of what will happen in vivo afterwards. Some of the studies, some of these systems are like one of the first invented was the microflux from Pion, which together with the microflux, which is conceptually similar, but the volume is smaller. They are based on the Pampa barrier, or other methods like the distoflux from Electrolab, where you actually have an insert that you can set into a standard permeability dissolution, pharmacopoeia dissolution setup, or the recently introduced the Permeter from Logan Instrument that actually also use the PermePad GIT, so the PermePad barrier. Another type of system that I have been developing lately is the so-called mesofluidic chip permeation assay. This is still on the, let's say, the development scale. So we combine basically a mesofluidic chip from React for Life, so that is called MEVO, with the PermePad barrier. And the advantage of this system is that instead of having a static system in the receptor media, like all the system we showed before, in this case, we have a chamber, a relatively small volume. And once the drug is permeated, we have a fluidic system in the receptor media. This has the advantage that removes completely the unstable water layer. from the acceptor, from the donor side, and also we can steer in the donor media, so reducing the unsteered water layer effect that in some cases affect the permeation. And through this system, we actually could investigate different solid dosage form and try to interpret the permeability, which of this formulation could have been the most promising to carry it out. We have investigated hydrocortisone because this is non-toxic BCS class 2 drug, so poorly soluble but medium permeable, and which is known to be subjected to supersaturation when formulated together with hydrophilic polymer. For example, in this case, using PEG 6000, we can actually achieve an increment in solubilization of almost double for the hydrocortisone. So we formulate hydrocortisone with the three different excipient, HPMC, PVP, or the crystal of hydrocortisone, and we co-freeze dry this sample. And we then dissolve this solid dosage form into the MIVO, measure the permeability over time. And what we have observed is that actually, according to what is reported in the literature, the HPMC, due to a combination of very high surface area and maintainment of the supersaturation, provided the best formulation possible for hydrocortisone. So these results show, compared to literature, were very good in agreement with the literature available. On the other hand, we also, with the same apparatus, we demonstrated, for example, the inability of regenerated cellulose to be used for a pH gradient experiment. We demonstrated this utilizing ketoprofen, that is a BCS class 2 drug, which is poorly soluble, but its solubility is dependent on the pH. But of course, instead of being also the solubility, also the permeability is dependent on the pH, because the compounds, in theory, should be more permeable in acidic pH, because in this case it's not ionized. And when we tested the ketoprofen using regenerated cellulose, we actually we could not see that. We just observe an effect of the permeability that was linked to the solubility of ketoprofen. So in this case, at pH 7.4, ketoprofen was much more solubilized. And what actually we saw is that that actually utilizing the permeaped, actually we got a reading of permeation for ketoprofen that was in line with what expected. So the permeability at pH 7.4 was lower that at pH, that acidic pH, and this is according to the literature, where this class of drugs is absorbed nicely in the stomach and in the upper intestine. So this is also an interesting approach, but that I would anyway classify into the dissolution permeation setups. And with this example, I actually conclude my presentation of today. I hope I could give you a nice overview on the cell-free biomimetic system available for analysis of interpretation of biopharmaceutics performance of drugs and solid dosage forms. And I would also underline that this work is some of the part of this work has been carried out here at University of Oslo, Department of Pharmacy, where I actually am working. And with this, I conclude. I thank you all, and if there are questions, I'm very happy to answer.
Thank you very much, Max. I think there was one question already from Girish. He asked if this new system is commercially available.
Which one? The
MCPA.
This is not commercially available, but it's you can assemble by yourself. You just have to buy the barrier and the MIVO, and then you can assemble it by yourself.
You can both request through Fabiok. It's not a problem. Just contact us through info@fabiok.com. Any more questions? I think what we always consider or offer to interest parties is that we have a specialized meeting together with Max to discuss the special application of you. So it's not a problem to arrange a specific meeting to discuss the problems or ideas. So we are very happy to help you to get your experiments right.
Absolutely.
For the next webinars, we will have one in November and one in December. So there will be 2 more Permeability series with exciting topics. I think one graphic was already shown today. I'm very excited to see the full scale of the study, and we will inform you on LinkedIn with all the information of the next Permeability series. And I will give the handout free, so you can download them now. Yes, you should see it. If any more questions arise, you can contact us through this email.
Yes.
All right. If there are no more questions, thank you very much for your attention. Max, thank you very much for your time.
My pleasure. Thanks a lot.
And your work. And please have everyone a good day or good night. Some of the Asian parties in Japan is already also here in the chat room. Good night. And looking forward to the next webinar. Bye-bye.
Bye-bye. Thank you.